Imagine a world where I could edit my own DNA. I could make myself immune to diseases, give myself superhuman powers, and make myself live for thousands of years. Well, this reality may actually be closer to us than you might think. And it's all thanks to a new technology called CRISPR gene editing. So what is it? How does it work? And why is it so important? Come, I'll show you. While it may sound like a chips brand, CRISPR is actually the name of a natural defense system found in bacteria. Bacteria fight against invading viruses by snatching bits of their DNA and storing it within their own genome, forming a sequence of clustered, regularly interspaced short palindromic repeats, or CRISPR. The bacteria can use its newfound knowledge of the viral genome to employ a certain enzyme called CRISPR-associated protein 9, or Cas9, essentially as a molecular hitman to hunt down the virus. Cas9 is given a guide RNA copy of the viral gene, which it can use to identify a virus. If the same virus attacks again, Cas9 will detect its genes and slice them at a precise location, neutralizing the threat. So how does this have anything to do with editing genes? Well, scientists realized that by replacing the guide RNA the cell gives to Cas9, we could reprogram it to target and remove any gene we wanted to. To give you an example of how we can use this, let's send Cas9 on a mission to genetically engineer and rescue a human embryo, a 2B person, from cystic fibrosis, a disorder that wreaks havoc in a person's lungs, caused by a single genetic defect. We can send Cas9 into the embryo, reprogram to take out the defective gene, which it'll do with great precision. Now, here's where things get real interesting. The cell will demand urgent repair of the gene. We could leave it to the cell to patch up the gene on its own, but without some sort of template, it's bound to mess up. But what if we sent in a modified copy containing a healthy gene? This way, the cell can use the modified copy as a template to repair the gene, not knowing it's been fooled into replacing it as well. As long as the surrounding genes in the copy are the same, the cell can't tell the difference. The mission is a success, and all it really took were 3 basic steps. Remove the old gene, send a template containing the new gene, and the cell does the rest for you. This same process can, in theory, be applied to edit any gene for any purpose. Now, does this mean we can all get superpowers? Not yet. CRISPR still isn't perfect, and there's still plenty of stuff we have no clue how to do. But even if we could, you have to ask the question, should we? I'm Ethan Xiong for the Breakthrough Junior Challenge.